connective tissues, making up approximately 1/3 of all our body's proteins and providing structural support for cells. In this sketch, we'll be heading to the Galapagos Islands to learn about collagen and how it's made. And don't worry about bringing your bathing suit and snorkeling gear because here at Sketchy, we got you covered. Now as you sip on that refreshing pina colada, let's start by going over what collagen looks like. This is definitely something you should know for exams. The basic structural unit of collagen is the triple helix, represented by the masts on the ship. The triple helix consists of 3 intertwined alpha chains. Each alpha chain consists of repeat segments of glycine, proline, and lysine. To remember the relative positions, we've drawn in the name of the ship, HMS Grasshopper, on the very left, a friendly shrimp-loving pro-captain in the middle, and a lizard on the right. Another boring fact is that 1/3 of collagen contains glycine. Why? Because it's small, just like the grasshopper, and can pretty much fit anywhere. Okay, before we go over the actual process of making collagen, let's review the 4 main types. Type 1 is the most abundant form of collagen, found in bone, skin, tendon, fascia, dentin, cornea, and sclera. Let these bone ores and that giant one rock in the background remind you that type 1 collagen is the primary type found in bones. Besides having the highest tensile strength, type 1 collagen is absolutely essential during the last stage of wound healing, specifically making scar tissue. Furthermore, we'll make this bone ore look brittle to remind you of osteogenesis imperfecta, sometimes called brittle bone disease. Type 1 collagen defects are usually seen with OI. Watch our congenital bone disorder sketch in Sketchy Path to learn more. Okay, let's mention type 2 collagen, which is present in cartilage, the nucleus pulposus of intervertebral discs, and the vitreous body of the eye. The key thing to remember here is cartilage. To highlight that, we'll draw 2 shark fins poking out of the water. Just a side note, sharks are cartilaginous fishes. They're also very terrifying, so let's get out of here. Phew! That was close. Type 3 collagen is found in reticulin, a supporting mesh for tissues. We can find it inside blood vessels, uterus, fetal tissues, and the pleuroperitoneal lining. We made this 3-shaped vine bloody red to help you remember the association of type 3 collagen and blood vessels. And one more thing, type 3 collagen defects are seen in the rare vascular Ehlers-Danlos syndrome. To remember this, just think of these fallen red Ehlers antlers. Alright, we'll wrap up this section with type 4 collagen, present in basement membranes, which serve as a point of attachment for epithelium. It's also present in the lens of the eye. Naturally, we selected Charles Darwin for this particular scene. He's inspecting the ground, which is just like the basement membrane of the Earth, with a 4mm lens. Type 4 collagen defects are seen in Alport and Goodpasture syndrome. Alport syndrome is a genetic disorder that causes eye abnormalities, sensorineural hearing loss, and glomerulonephritis. To remember Alport syndrome, think of this port sign. In contrast, Goodpasture syndrome is an autoimmune disease that results in disruption of type 4 collagen in glomerular and alveolar basement membranes, leading to rapidly progressive glomerulonephritis and pulmonary hemorrhage. Goodpasture syndrome is represented by this grassy pasture. Watch our nephritic syndrome sketch from Sketchy Path to learn more. Alright, let's change gears and go over the process of making collagen. The process starts in the usual way, with transcription and translation. Inside the nucleus of fibroblasts and other collagen-producing cells, collagen DNA is first transcribed into mRNA before getting transported out of the nucleus. Next, alpha chains are partially translated by free ribosomes. The signal sequence at the N-terminal end of the newly synthesized polypeptide binds to a signal recognition particle on the rough ER to direct the polypeptide into the rough ER. Once inside, the signal sequence is removed and ribosomes finish translation to produce pre-procollagen. We'll draw this rocky shoreline to show the rough ER. Here, pre-procollagen undergoes post-translational modifications. The first modification is hydroxylation of proline and lysine residues with help from vitamin C. These dang lizards are everywhere. Poor sailor was just trying to enjoy his orange and ward off scurvy. This step is absolutely essential for creating a stable triple helix. And speaking of scurvy, vitamin C deficiency causes scurvy due to impaired collagen synthesis. Watch our vitamin C sketch to learn more. The next step is adding glucose and galactose monomers to hydroxylysine residues, also called glycosylation. This is represented by the hexagon-shaped chain keeping the rascally lizard captive. After these modifications are done, 3 alpha chains twist around each other to form a triple helix held together by hydrogen and disulfide bonds. Shown here is a sailor braiding 3 ropes to make a triple helix. And just a heads up, we're now calling this procollagen, and it will go to the Golgi for sorting. Afterward, procollagen gets packaged into a vesicle to be transported to the extracellular space via exocytosis. Exocytosis is basically like this raft carrying the braided rope back to the ship. Just imagine all those tiny little rafts floating around your body all day, bringing rope from shore to ship. Pretty profound stuff, actually. Before we continue, just know that everything that happens next takes place in the extracellular space. Collagen peptidases remove the disulfide ends of procollagen to make insoluble tropocollagen. This diligent sailor cutting the ends of the braided rope is collagen peptidase. That's some nice ASMR if you're into that. Anyways, we're almost done here. Let's go to the final step. Here, lysyl oxidase uses copper to cross-link tropocollagen molecules via hydroxylysine and lysine covalent linkages, finally creating collagen fibrils. To illustrate this, this tireless sailor is adding copper C-rings to connect the braided ropes. And to conclude, we'll draw this chaotic monkey going bananas on the C-rings. The monkey will remind you of Menkes disease, an excellent connective tissue disorder that results in disruption of copper absorption. Collagen cross-linking is impaired in Menkes disease. As much as I want to keep monkeying around, unfortunately, it's almost time to go. But before we sail away, a quick summary. Collagen is a major structural protein found in connective tissues. Collagen is composed of a triple helix which contains 3 alpha chains of glycine, proline, and lysine repeats. There are 4 main types of collagen. Type 1 is present in bone, type 2 in cartilage, type 3 in blood vessels, and type 4 in basement membranes and the lens of the eye. Collagen defects are seen in OI, vascular Ehlers-Danlos, Alport, and Goodpasture syndrome. After making pre-procollagen, it undergoes post-translational modifications in the rough ER. First, proline and lysine residues are hydroxylated using vitamin C. Next, sugar groups are added to hydroxylysine residues before it goes to the Golgi for sorting and leaves the cell via exocytosis. In the extracellular space, collagen peptidases remove the disulfide-rich ends of procollagen to make tropocollagen. And finally, lysyl oxidase uses copper to cross-link tropocollagen molecules, forming collagen fibrils. Collagen cross-linking is impaired in Menkes disease.